Welcome back to Beyond 101. In the last video, we discussed three pathways for complement system activation. So recall that for activation, we have the classical, alternative, and lectin pathways. And the downstream effects of the complement system are threefold. The first is going to be, remember, opsonization. The targeting and signaling for phagocytosis. The second being inflammation. And the third being membrane attack. In the last video, we discussed opsonization pretty thoroughly, but we didn't really give inflammation or membrane attack the time of day. So in this video, we're going to be concluding our discussion of the complement system by discussing inflammation, membrane attack, and the ways in which we can regulate all of these processes. Let's start off with membrane attack, and membrane attack that pathway picks up right where we left off in our complement activation pathways, with a functional C3 convertase. And remember that if we're talking about the classical or lectin pathway, that C3 convertase is going to be our C4b2a C3 convertase. And if we are in the alternative pathway, it is going to be in the form of C3b B. And recall that the point of these C3 convertases is to convert C3 into C3a and C3b. Well, the next step of this pathway is going to be using some of the C3b that we made, and it is going to combine with our C3 convertases. And it is going to make one of two products, depending on the C3 convertase we started with. If we are starting with our C4b2a C3 convertase, then we will form what is known as C4b 2a 3b. And if we are starting again with our alternative pathway C3 convertase, then we will form what is known as, right here, C3b2 BB. Both of these are known as C5 convertases. And it shouldn't take a rocket scientist to figure out what a C5 convertase does based off what we know about what C3 convertase does. C5 convertase converts C5 into, it's going to break C5 up into C5a and C5b. C5a is very similar to C3a, but we could sort of think of it as C3a on steroids, because C5a is a major inflammatory protein, meaning that it can cause significant inflammation, whereas C3a causes moderate inflammation. And in addition, just as what we saw with C3a, it is we can think of it as like a dual authenticator for phagocytosis. And both of these processes we're going to discuss with more detail later in the video. But in the case of our membrane attack pathway, we're going to focus on C5b. So C5b is going to sort of break apart from its convertase, and it is going to combine with two other complement proteins. So if we have C5b over here, it will combine with two new complement proteins. It is going to combine with the complement protein C6. This is C6, and it is going to combine with the complement protein C7. And C7, once it binds, is going to undergo a conformational change that allows for this whole complex, which I will illustrate right now. It is going to become the C5b, C6, C7 complex. And it is this conformational change, and I'm going to draw it, move this label down, because it is this conformational change that allows for C7 to integrate itself into the microbial membrane. So if we have a phospholipid, then we will see that our new complex is going to be able to burrow itself in. And this allows for the next event, which is going to be introducing our next complement protein, which is C8. And C8, similar to what we saw with C1, actually is made up of 3 parts, but 2 of them are going to be grouped together. And that's what we're going to see in this large fragment. This is going to be C8 alpha gamma, and our small part is going to be C8 beta. And it is C8 beta that is going to actually bind with C5b. And this is going to allow for C8 to fully ingratiate itself into the microbial membrane. So if we draw out our complete complex as what we have so far, here's the microbial membrane. Here is our C5A, C6, C7 complex, and C8 is going to nestle itself in right here. And it is this process that is going to truly initiate the membrane attack portion. And that's done with our last complement protein, which is known as C9. And as a matter of fact, we're not going to just have one C9. We're going to have anywhere from 10 to 16 C9 subunits. And these are going to form a pore once they bind with C8. And it will look something like this. This is what we call the membrane. So membrane attack complex, or MAC. And the membrane attack complex essentially is a pore where the innards of whatever the membrane attack complex is bound to can start to leak out. And this is going to be how membrane attack works in the complement system. And it's important to note that although this is a pretty, spectacular display, it's actually quite rare, and it doesn't contribute much to the overall effectiveness of the complement system. This is a terminal stage of the complement system. Most of the effectiveness of the complement system happened all the way back when its activation with the formation of C3 convertases. But this is the end product of the complement system. This is what we've all been building up to. The last function of the complement system is to promote inflammation, and this is done by the small fragments of our complement proteins. So specifically, this is going to be done by C3a, C4a, and C5a. And recall that C5a is our major inflammatory protein, meaning that all the effects that we discuss in this section are going to be caused most strongly by C5a. But what are those effects? The first and most important effect is that these 3 proteins lead to increased vasopermeability. What is vasopermeability? Vaso refers to blood vessels. And permeability refers to the idea of moving some sort of substance across a membrane. So moving across a membrane / barrier. So in the case of vasopermeability, if we're increasing it, it means that the things inside of the blood vessels can now move outside of the blood vessels easier. And I think a good way to demonstrate this is through a diagram. So I'm going to draw out right here what a blood vessel might look like. So the blood vessel has a wall that surrounds it, and the wall is made up of these cells. And these cells are known as endothelial cells. Inside our blood vessel, we'll find things like red blood cells, we'll find things like our white blood cells, our immune cells, and we'll also find other proteins, sugars, other sort of macromolecules. However, if we have an area of infected tissue, that's going to obviously be on the other side of this wall. So if we have these green cells, which are meant to be our infected cells, they're going to be on the other side of the vessel. So by increasing vasopermeability, we make it easier for the white blood cells and other proteins, for example, other inflammatory agents, maybe antibodies or other complement proteins, to move from inside the blood vessel to outside. The second main function of these small fragments specifically has to do with C3A and C5A. So C3A and C5A, they activate receptors, specifically ones known as G protein coupled receptors or GPCRs on immune cells, and the activation of those receptors leads to secretion of other inflammatory agents. So a very notable example is in mast cells. So C3A and C5A will activate G protein coupled receptors on mast cells, and this leads to the secretion of other inflammatory agents. So secretion of inflammatory and the two that are associated with mast cells, one of them is known as histamine. And anyone with a pollen allergy will be familiar with the idea of histamine, because in the springtime, you might take a Zyrtec or a Claritin in the morning to try to alleviate your allergic symptoms. And the reason why those drugs work is because they're antihistamines. They reduce the effect of histamine on the body. And then the second protein is one that is known as TNF alpha, which stands for tumor necrosis factor. This is a protein known as a cytokine, and it's something that we're going to dive into much greater depth later within this series. The next function has to do with C4A. And C4A's function is slightly different than C3A and C5A because the GPCRs that the immune cells have don't respond to C4A. However, C4A is what connects complement to coagulation. I hope I spelled that right. Coagulation in this context referring to the clotting cascade. Clotting is what happens when our red blood cells, cluster together. This is a crucial part of the healing process, and by agglutinating and coagulating all of these components together, it allows for the slowing down of blood flow and the accumulation of potential immune cells and other immune elements. The last function of these small fragments kind of ties back to the first in the idea of moving cells across the walls of blood vessels, and it's that the cells that make up the blood vessel walls, the endothelial cells, and our immune cells will produce what are known as adhesion proteins. And these adhesion proteins allow the white blood cells to stick better and easier to the walls of the blood vessels, which makes it easier for those white blood cells to then move across the barrier. So this leads to increased ease for immune cells to cross from inside vessels to outside. So all of these functions have the overall effect of increasing, so all four increase the recruitment of immune cells and proteins to the site of infection. In its totality, the point of these small fragments is to make it so that immune responses can happen quicker and more efficiently. So we spent the last video and a half discussing how the complement system is activated and what its downstream effects are. But what's equally important is understanding how it's regulated, because if the complement system is allowed to spiral out of control, to increase exponentially without any sort of limit, or if it's not able to be turned off after the infection has been properly dealt with, we can see some really nasty effects. And as you can see here, we have a bunch of different proteins found in various locations that will serve different functions. The first one we're going to discuss is one that is known as CD35. CD35 is also known as decay accelerating factor, or DAF. DAF is found on cell membranes and its mechanism of action is that it binds C3b and C4b. And what this leads to is it will break C3 convertase. It breaks it apart back into its C3b and BB components, or C4b and C2a components. At the same time, because CD35 will bind to C3b and C4b, it can prevent the C3 convertase from forming in the first place. So it prevents its formation. In a very similar mechanism of action, we have another protein known as CD55. CD55 is also known as CR1. And CR1 is going to come back later when we discuss phagocytosis, but in the context of complement regulation, it's going to perform a function very similar to CD35. So it's also found on the cell membrane. And to be clear, it's the cell membranes of host cells, specifically of macrophages. And as I said, CD55's function is the same as CD35. It will bind to C3b and C4b to prevent the formation of a C3 convertase or to break apart an already existing one. Moving on, our next regulatory protein is known as C4 binding protein. Which can be shortened to C4BP. Now, unlike the ones we've discussed already, C4BP is not found on the cell membrane. It's found circulating within the blood, so within circulation. And its function is going to be, again, similar to what we saw with CD35 and 55, but instead of it binding C3b as well, it's only going to bind C4b. So it breaks C2a from C4b. Again, preventing the formation of a C3 convertase, or breaking the C3 convertase itself. Moving on to our next plasma protein, we have what is called factor I. Factor I, again, found within circulation, within it's plasma soluble, so I'll say again, within circulation. And factor I is going to have a different mechanism than what we've discussed so far, because factor I is a protease, and specifically it's a C3b protease. Factor I can cleave C3b into what is known as IC3b. And we have another product which is known as CFB. And IC3b cannot form a C3 convertase. It is entirely non-functional in the context of making a C3 convertase. It has other functions which we'll discuss later, but IC3b will never be able to go through the alternative pathway to make a C3 convertase. Because this is such a powerful tool, it makes sense that there's going to be another form of regulation for factor I. And that form of regulation is that it requires what is called a cofactor. Make a little space here. And cofactors, again, recall back to intro biology, are things that can bind to an enzyme on a place other than the active site and increase the enzyme's function or allow the enzyme to function properly. So cofactors for factor I include CR1, which we already discussed, C4BP, which we discussed already, and two new ones. The first new protein is known as CD46. And CD46 is also known as MCP, which stands for membrane cofactor of proteolysis, which means the breaking of a protein. And the last cofactor that we haven't discussed yet is one that is known as factor H. So this is all the ways that we can regulate C3 convertase. But there are other targets for regulation, and it makes sense that the majority is for C3 convertase because, again, C3 convertase is the converging protein for all of our pathways of activation. C3b is crucial for the formation of a membrane attack complex. Remember, it's the first step, and C3b is our main opsonizer. So it makes sense that regulating the formation of C3b through our C3 convertase is a very powerful way of regulating the complement system. But moving on to non-C3b related targets, we have what is known as C1INH, which we could think of as short for inhibitor. C1INH is found within circulation and as the name might suggest, it targets C1. And how it works is that it will irreversibly inhibit C1R and C1S. Recall that these are our proteins that will break C4b and C4 and C2a in order for them to bind to form a C3 convertase. So this is effectively preventing the C3 convertase from forming through the classical pathway. The last protein that I'm going to mention in this video is one that is known as CD59. CD59 is also known as protectin. Protectin is found on cell membranes and its job is to bind C8 to prevent the formation of membrane attack complexes, to prevent MACs from forming. Remember that C9 binds to C8, and we have a bunch of the C9 proteins, and they form the pore. So if we bind C8 and prevent that process from happening, then we effectively eliminate the formation of membrane attack complexes. So this is not an exhaustive list of all the different regulators of the complement system, but this will give us a pretty good idea of how the complement system is regulated. The last part of this video is going to be focused on phagocytosis, and we've mentioned phagocytosis and its regulation a few times throughout the last two videos, but I want to use this time to go through it more rigorously. So let's imagine that we have a pathogen that has been opsonized by C3b. It will look something like this, where it will have C3b on its surface. And now let's imagine we have some sort of cell that can perform phagocytosis. Maybe it's a macrophage, maybe it's a neutrophil, but as we know, these cells will be covered with a few receptors, but the one that binds C3b is the receptor CR1. And as we've mentioned, binding of C3b to CR1 is not sufficient to cause phagocytosis. So detection of C3b by CR1, CR stands for complement receptor, so complement receptor 1, will not cause phagocytosis. As we said, we need a dual authenticator, and in the case of the complement system, that's going to be the small fragments of our complement proteins, that being C3a, and the new one we mentioned today, C5a. And we're going to focus in specifically on C5a because there's a receptor on the surfaces of macrophages called, and I'm going to draw it here, this receptor is known as C5a R1. And C5a R1 will binds C5a, and it will cause phagocytosis. When coupled with the CR1 C3b binding event. There's another receptor that's worth mentioning that will recognize C5a, and that's the receptor, and I'm going to draw it in this lighter color. This receptor is known as C5a R2. And C5a R2 is interesting because when C5a binds to C5a R2, nothing really happens. So the prevailing understanding is that C5a R2 almost acts as a dummy receptor, and this is important because it's another way of limiting phagocytosis in scenarios where we might not want it to happen, because if a cell accidentally gets tagged for phagocytosis, again, with this dual authentication system, you need C5a to bind to a receptor, and if it binds to C5a R2 and the phagocytosis event wasn't valid in the first place, then that's another way of preventing unnecessary cell damage. However, this axis with CR1 and C5a R1 is not the only way to initiate phagocytosis, because, as we know, if we have a cell that is covered in, or a pathogen, I should say, that is covered in C3a and binds a macrophage that has CR1, we have the possibility of another cleavage event happening, that being with the presence of what we just discussed, our factor I. Because recall, CR1 is a cofactor for factor I, and if factor I binds to C3b in conjunction with CR1, we get the formation of our new IC3B. So IC3B, again, will not lead to the formation of a C3 convertase. However, it's not entirely useless because there are a series of receptors that can bind IC3B and lead to productive results. These receptors are CR2, complement receptor 2, which is also known as CD21, complement receptor 3, which is also known as CD11B, CD18, CR4, which is also known as CD11C, CD18, CD18, and a receptor called CRIG. Of these that are important to note are CR3 and CR4, because CR3 binding and CR4 binding to IC3B is enough to cause phagocytosis on its own, so without needing the C3B to bind to anything. So it will cause phagocytosis, or I should say causes phagocytosis without dual authentication. CR2 is a little more interesting, so I'm going to make some space for it on the side, because CR2, it binds more than one ligand, so binds more than one ligand, and its location is also somewhat interesting. It's found on B cells, and B cells are the primary producers of antibodies. But let's go into this more than one ligand idea, because CR2 can bind IC3B, but IC3B can undergo another cleavage event. If IC3B is bound by factor I and an appropriate cofactor, IC3B can actually be cleaved into two new products. One of them is known as C3C, and the other is known as C3DG. C3C will dissociate. Specifically, will dissociate away from the surface of whatever it was the C3B was originally coding. But C3DG will stay on microbial surfaces that are a little bit messy, surfaces, and is detected by CR2. And remember that CR2 is found on B cells. So when CR2 is activated, it leads to increased, so binding leads to increased B cell response, including increased antibodies. That just about wraps up this video. We discussed how membrane attack occurs through the formation of membrane attack complexes. We discussed how the complement system leads to inflammation. We discussed how the complement system is regulated, and we discussed how the complement system starts the process of phagocytosis. And in the next video, we're going to finally be moving on to the cellular components of innate immunity.